Hi, I'm Hayden Connery.
I'm Julia Miller.
I'm Lily Smith.
I'm Cassie Miller.
I'm Rick Stockmeyer, and we're Group 6, presenting Animals, Genetic Clues to How Dogs Became Man's Best Friend.
Our article focuses on the domestic dog's road to domestication by analyzing genes and hormones that are believed to play a role in dogs' sociability and communication skills with humans. More specifically, genome sequencing data propose that behavioral traits influenced by genes linked to neurocrest signaling pathways fostered the initial selection process during early domestication of dogs. Animals with fewer neurocrest cells secrete less adrenaline and are more comfortable around humans. If this animal is bred, it would pass this behavior onto its offspring. Neurocrest cells also form parts of the face, control the color of the skin and hair, and form cartilage in the ears. This is why a domesticated animal with fewer neurocrest cells would have floppy ears, a shorter snout, and splotchy fur. Additionally, the article explains that, quote, a genome-wide polymorphism analysis of a global sample of dogs and wolves suggests that there had been selection pressure on the adrenaline and noradrenaline biosynthesis pathway, well known for its involvement in the fight or flight response. Other genome-wide analyses have also identified various genomic regions as candidates for affecting dog cognitive phenotypes, such as inhibitory control, sociability, and interspecies communication, end quote. Tanoike et al. 2022.
The article recounts two tests that evaluated the human-related cognition ability of the researchers' sample size of 624 domestic dogs based on Parker's cladogram. This cladogram divides these dogs into 23 different clades represented by the 23 cookie bits. The article then further divides these breeds into two groups. The ancient group includes the dogs that underwent the first domestication, and the general group includes the dogs that underwent the second domestication of modern dog breeds. While there wasn't a normal difference in efficiency, the ancient breed appeared less reliant on human assistance, whereas the general group looked at the experimenters much more. Furthermore, studies found that factors such as sex, training, and neutered status had little effect on the outcome.
Okay, so in order to go from our ancient breed to our general breed, the researchers found a few differences in the cortisol and oxytocin levels. So here we have our oxytocin hormone, also known as the love hormone. And in the general breeds, we saw an increase in the amount of oxytocin levels, and this led to the dog-human bond and the friendliness that we see now in our general breeds. And here we have our cortisol, and in the general breeds, we saw decreased or regulated cortisol hormone levels, and this led to a decrease in the stress, the fight or flight response, and ultimately a lack of fear towards the humans.
Here we have our ancient and our general breed of dogs. The yellow beads represent the TT base pairs. The blue beads represent the C base pairs. And the green beads represent CT base pairs. In the WBS gene, you can see that the ancient breed has more TT base pairs, and this WBS SNP was synonymous and associated with the number of gazes to the human during the problem-solving test. Next, in the MC2R1 gene, we can see that the general breed has more TT base pairs, and the ancient has more CC base pairs. Lastly, we have the oxytocin receptor 4 gene. This ancient breed has significantly more TT base pairs, and this SNP led to an increased duration of glances and number of glances to the human during the problem-solving test. The next three base pairs we have are GG represented by yellow, AG represented by green, and AA represented by blue. First we have the MC2R2 SNP. As you can see, the general and ancient breed have very similar frequencies of each base pair. However, for the MC2R3, the general breed has more GG base pairs as compared to the ancient breed with more AA base pairs. In all MC2R genes, there was one non-synonymous and two non-synonymous SNPs. SNP 1 and 3 correlated to duration and number of gazes to the human for the problem-solving test, and SNP 2 correlated to an overall correct choice by the breed. In the oxytocin SNP, you can see that the general breed has more GG base pairs. The oxytocin SNP led to latency and gaze periods in the problem-solving test.
During our replication of the experiment, a treat was put under one of the two bowls that were presented to the dog, and the experiment was to indicate to the dog which bowl contained the treat using a point cue. The behavior of the dog was noted and whether or not they picked up on the cues that the experiment was giving them and successfully found the treat. This is Jacques, who represents the general group. It can be seen that he looks at the experimenters often and can't solve the problem as quickly. This is Roscoe, representing the ancient group. He is able to somewhat independently retrieve the treat from under the cup.
In conclusion, researchers found multiple SNPs on genes that are associated with the domestication of dogs. SNPs on the oxytocin receptor and WBSCR gene related to the increased friendliness of these modern breeds. These SNPs also related to the duration of the two-way test study and the number of gazes that the breed gave to the human during their choices.
Here we have chromosome 18PR. It holds the MC2R gene. The MC2R gene SNP2 relates to the breed selecting the correct choices in the pointing test, and SNP1 and 3 relate to increased friendliness. The oxytocin SNP related to the latency in the problem-solving test. Overall, it was found that the ancient breeds were less reliant on humans as compared to the general breeds due to the frequency differences of each of these SNPs.